So this is a PCR, also known as polymerase chain reaction. This is where our three steps actually happened. Denaturation, which happened in 95 degrees Celsius. We have annealing, which happened from 50 to 70. And also we have elongation, which happened at 72 degrees of Celsius. Yeah, this is where all those three steps happens actually. So as you can see here in the opening, you put your two reaction in test tube, which contain primer, buffer, polymerases, your template DNA, nucleotides, DNTPs, and DD deoxynucleotide DDNTPs. You have that mixed in your tube, and then you're gonna put in one of the holes here, small holes actually, and then you're gonna close it, set up the program, and then you're gonna run your PCR reaction. Usually it goes through cycles. Yes. So basically, what happened during the denaturation process is your DNA strand will separate, and during annealing, they will try to come back, and then during extension, it will just form more copies. So at the end of the day, what PCR does is to form more copies of your DNA for the future progression. So that's what about PCR. We have absolutely make a great step in the science field nowadays. We are able actually to combine two different DNAs from two different organisms together and form one thing in common. Not necessarily one thing that is gonna, but the point of combining two DNAs so that we can either do research, or we can learn about a certain gene expressions, and that's a great step in science. So today in my video, I just want to talk about the entire process of gene cloning. What happened? What's the process? For example, I mean, I want to produce insulin or some kind of chemical, but I have to get some of the genes of interest from different organisms, and I have to use bacteria. So how can I combine them together and ended up with my genes, or even suspect if there's a murder case right there, and I don't know, and I have to go through a procedure, do all the DNA sequencing, and at the end of the day being able to analyze the DNA and come up with a conclusion that who did that. What are the some of process that happen in the lab? That's what I'm going to talk in my video today. So part of my video has actually have the PCR machine, PCR polymerase chain reaction. Yeah, so this is a very important step during your process. So what PCR machine does actually is to make more copies of your gene of interest. So we actually need more copies to work on. So PCR does that, and it goes through three steps, as we have seen or discussed in our lectures. You have denaturation, which is a process whereby DNA template actually divided. They need to separate denaturation, yeah, and this is happen usually in a 95 degree of Celsius. Then the next step is annealing. It anneals under 55. Usually it's from 52 to 60. 9 if not 7, yeah, and then the last step is extension, whereby then the DNA come together and form those new strand together, and the temperature is usually under 72 degrees of Celsius. So in PCR this process will be going in cycles actually, to make those copies of DNA. So after we have our primers, and I've talked in my video, you will see the video I had. I actually went to the lab and actually see the PCR machine, and I've talked everything you needed to run the reaction, all the things that they are needed. But basically you need a buffer so that it can maintain a pH. You need a DNA template so that it can also you need primers to attach to that DNA template. Yes, and what else do you need? Yeah. Okay, so those are the things that you need for your PCR reaction. Then after you're done with your PCR reaction also, so after you're done with your PCR reaction, you need to run a gel And what's the point of a gel actually? You want to see if your PCR was successful or not. So jelly electrophoresis help us to see if our PCR was successful or not. If it was not successful, then that means we have to go back again and check in our primers DNA sequence what went wrong or what went right and do it again. But if it was successful, then we should be able to see those bands and we can continue or move on. So there's a procedure how to make a gel, and there's a things actually needed. So actually gel is a carbohydrate. It's a polysaccharide, and it comes in the form of powder. You mix it with the buffer and put it in a microwave. You heat it, and then after that, then yeah, you can obtain a gel, and you run it through some sort of electrical. I wish I had a picture electrical vessel, and usually it has two sides. It has a negative side and a positive side, and our DNA protein is negative side, so it will move from negative to positive based on the DNA molecular weight. So the bigger the DNA, the slower the movement. So the small ones will move fast, and the heavy one they'll move slow. So if you're about to look at the picture, actually after gel electrophoresis has been performed, then you'll see the differences between bands. You should be able to know, oh, this one is big, oh, this one is small, depends on their shapes. That's one thing. So gel will help us to tell us, okay, your PCR was successful, so now you can move on. So okay, now, and I'm sorry I didn't mention this earlier. Imagine we are dealing with GFR, which is green fluorescent protein. That's our gene of interest. We want to transform that thing to E. coli. E. coli is one of the bacteria that, yeah, so there are some things that we'll need. We'll need a plasmid. Plasmid, which is a form of DNA, actually, it's a vector. Vector is something that carries something. So vector, actually, plasmid is a vector that is carry this DNA, and it allows these two different DNAs to combine together. That's the point of genetic or DNA recombination. When you have two DNAs from two different organisms and combine them together for the sake of maybe biotech reasons, for the sake of research or whatever. So imagine we are dealing with the GFR, green fluorescent protein. So now what we need to do, we need to take that gene of interest actually and put it inside our vector. So we need to cut the vector, and just like the same that you will need a scissor to cut this paper. So even in plasmid, you will need some kind of cutter to cut that paper. So imagine this is our vector, and we need to and vector can come in a linear or circular way. And for my video, let's talk circular way. So scissors would be restriction enzymes. Yeah, restriction enzymes, they act like scissors, and you have different types. You have HINT3, you have RNPL, you have a lot of types of restriction enzymes, but the point of restriction enzyme is actually to cut. So you have restriction enzymes, they will cut the your vector. And usually they look at the DNA sequencing for the something called the palindromic sequence. They are very good at recognize, oh, this is palindromic sequence. Then they cut in that way, and then after they cut there, we go. Actually, this place is called So they cut it, restriction, digestion, digestion. So they digest it. After doing that, then you insert that gene of interest, and then you have to do ligation, because you can't just cut things and leave them open. You need to combine them together so that other process can happen. So after we cut our vector with restriction enzymes, then we perform a process called ligation. To ligate is to actually, if our plasmid was a cycle, we cut them, we insert the gene inside, and now we have to ligate, and actually have some things that you need in order to ligate. Then after we ligate, we need to clean up, because sometimes some of the buffers, some of the salt can affect our DNA, so we need to do a digestion cleanup with some alcohol or things like that, and then we can continue on. I forget to talk about then after that we have to do transformation. transformation on the plates. Yeah, so you have to make a gel on your plates. I don't know if you wanna use agarese or you wanna use LB broth, but for some reason, not sometimes, we need to use plates so that we can see our growth. There are some things that we need in our plate, things like X-Gal and IPTG. So what is X-Gal? X-Gal, this is a chemical that it's a chemical that it helps us to tell us if actually our bacteria have successfully receive the DNA, and if they have successfully received a DNA, then the colonies in the plates will be white in color. If they have not, then the colonies will be blue in color. So we want to see white because that means successful. Blue, that means the recombination didn't happen. So yeah, so X-Gal is good at doing that, and IPTG acts as a lactose. It's a inducer. Um, yeah, and I think you remember from Lac Open Run, you have some section there. You have a activator, you have a repressor. So IPTG come there and take repressor off, and actually allow the process of transcription to happen. So you have those two things that are very important. And once you know that, oh, okay, so our transformation has happened, and I told you E. coli, they're very useful to use them as a transformation.